Hi, and good evening. Thank you for joining us today to what we're jokingly calling Nashville 2017. My name is Dr. Lorraine Rush, and I'm president at High Point Clinical Trial Research. We're very pleased to share that we have over 300 attendees registered from North America and Europe joining us today for this topical survey of NASH drug development. The high interest in this area is a result of the race to a therapy for NASH, which, as you all recognize, is a complex disease involving multiple progression pathways we're striving to understand as an industry. The format of today's webinar will be short presentations followed by brief Q&A. Please electronically submit your questions during each presentation. We have deactivated the raise hands function due to the number of attendees, but we'll do our best to answer as many questions as possible and have allotted additional panel time to address questions at the end for those able to stay on the line. This format was developed to allow for a jump on and off type of attendance for people with an interest in one specific area or to use as a general overview. Finally, we'll make the audio version of this webinar available to our attendee list. It's also our plan to develop a fact sheet which addresses all of the questions asked in the forum, regardless of whether or not we were able to take them due to time constraints. This will be sent to the attendees along with the links to the presenters for direct outreach. The order of today's speakers will be Dr. Sabina Paglialunga from Celerion, Tim Montgomery from Metabolomics, Dr. Nunez from High Point Clinical Trial Center, and Dr. Michael Murphy from Pacific Biomarkers. Speaker number one, as I mentioned, will be Dr. Sabina Paglialunga. She is the Associate Director of Scientific Affairs at Celerion. Sabina has a strong background in the metabolic disease space and functions as the Associate Director of Scientific Affairs at Celerion, where she acts as the liaison for clients in need of subject matter expertise. She is here to provide an overview on the new therapeutic development of NASH medicines. The focus of her discussion will be the current understanding of the disease, the challenge of diagnosis, and an update on the development landscape. And with that, I'd like to turn it over to Sabina.
Thank you, Dr. Rush. So we'll begin with non-alcoholic fatty liver disease, which encompasses the entire spectrum of fatty liver in individuals without significant alcohol consumption, ranging from fatty liver or steatosis to cirrhosis. NAFLD is a chronic metabolic disease which is histologically characterized. So fatty liver or steatosis is identified as more than 5% of fat within the liver. Non-alcoholic steatohepatitis is steatosis with inflammation and injury. And then NASH with cirrhosis is the most severe form of this disease. NAFLD is often asymptomatic, and patients can have normal liver enzyme function tests, making it very challenging for early diagnosis. It is estimated that approximately 25% of the general population will have fatty liver. Of those with steatosis, approximately 60 will progress on to NASH. And currently, there is no biomarker able to predict those who will progress onto more severe forms of the disease versus those who will remain in a more benign-like fatty liver stage. 30 to 40% of NASH patients will develop fibrosis, and 15 to 20% can progress on to cirrhosis, which can lead to end-stage liver disease, transplant, or hepatocellular carcinoma. There's also a small percentage of subjects that will develop hepatocellular carcinoma independent of fibrosis. However, more research is required to understand and determine the incidence of this phenomenon as well as the underlying cause. The following graph shows the trend in new liver transplant wait list registrations from 2004 to 2013. And as you can see, NASH has demonstrated the greatest increase. And NASH right now is even the second leading etiology for liver transplant. Within the next few years, it's anticipated to become the first due to the rising prevalence of the disease, as well as the fact that there's a decline in hepatitis C-related cirrhosis with new antiviral therapies. NASH is highly associated with metabolic syndrome, and risk factors for non-alcoholic fatty liver disease include type 2 diabetes and obesity. An estimated 70% of NAFLD patients will have type 2 diabetes. Other risk factors include advanced age and ethnicity. And research has shown that there's a prevalence of NASH within the Hispanic and Asian populations. This may be linked to genetic factors. Genetic variants in adiponutrin, or also known as the gene PNPLA3, are highly associated with NASH progression. And then there are dietary factors. So excess fructose consumption can lead to an upregulation of de novo lipogenesis, which is the new synthesis of fat. This, coupled with a diet high in fat, can lead to accumulation of lipids and triglycerides within the liver. And this is a global phenomenon. Worldwide, there is a strong correlation between the prevalence of obesity and the prevalence of non-alcoholic fatty liver disease. So how do we get from a healthy liver to a cirrhotic liver? I'll walk you through what the current line of thinking is. This is a simplified schematic showing the heterogeneity of the liver and the different cells found within the liver. So there are the hepatocytes, which are the main cells, and they're involved in protein, cholesterol, and triglyceride synthesis, as well as drug metabolism. There are also monocyte-derived macrophages, as well as Kupffer cells, which are stellate macrophages. And in the liver, there are also hepatic stellate cells, and these are a quiescent dormant type cell. Dietary fat, fatty acids from adipose tissue lipolysis, de novo lipogenesis, all contribute to fat accumulation within the liver. Insulin resistance promotes a lipotoxic environment with increases in pro-inflammatory cytokines, and this can result in lipid-induced injury, which promotes endoplasmic reticulum stress, apoptosis, necrosis, and inflammation. Fibrogenic cytokines such as TNF-alpha, TGF-beta, interleukin-1 can stimulate hepatic stellate cell transdifferentiation from a dormant cell to an activated fibrogenic type cell. And these activated cells can proliferate, increase fibrogenesis, collagen synthesis, extracellular matrix production, all resulting in hepatic fibrosis. At the other end of the pendulum, we have fibrosis regression. This can be achieved through reducing the number of activated hepatic stellate cells, lowering extracellular matrix production, and upregulating fibrolysis. So these activated hepatic stellate cells can have three fates, and they are to enter into a senescent state of growth arrest, where they are cleared by immune cells, converted to an inactivated form, yet remain primed to revert back to a fibrogenic producing cell, or undergo degradation through apoptosis. And fibrosis regression is a natural process of cellular repair, and this will typically occur in the liver upon acute liver injury. This system, however, can be disrupted with chronic disease such as NASH, and therefore would require intervention, therapeutic interventions, for fibrosis regression and resolution. Now, to diagnose NASH, this is done by liver biopsy and histological assessment, and this is currently the reference standard. A NASH diagnosis is based upon a graded score, the NAFLD activity score, and it has three components: steatosis, lobular inflammation, and ballooning injury. A NASH score of 4 or more is indicative of NASH. Not a component of the NASH diagnosis, however, it is important to rate the severity of the disease. So fibrosis staging qualifies the fibrosis pattern and progression towards cirrhosis from F0 to F4. And each stage is approximately 7 years. there is a subpopulation of rapid progressors that can develop cirrhosis. The main challenge with the NASH indication is the need for liver biopsy to diagnose the disease. And liver biopsy is not a feasible long-term approach for monitoring the disease diagnosis or assessing treatment response. The liver biopsy itself reflects 0.002% of the entire liver. It's also an invasive procedure with low acceptance patient rates, risk of complication. It can be afflicted by sampling error, and it's not appropriate for monitoring the disease on a short-term basis. So there's definitely a need for non-invasive approaches. There are a number of non-invasive techniques in development to diagnose NAFLD, which include soluble biomarkers and fibroscan magnetic resonance. And we'll hear more about these various modalities in the upcoming presentations. One technique that I do want to highlight is magnetic resonance imaging. And here, hydrogen atoms from water and those from fat will have a different properties under a magnet. So the amount of fat fraction, water fraction, or the proton density fat fraction can be determined. And using this technique, it's been shown to have good separation among the 3 histological grades of steatosis, as well as good sensitivity and specificity compared to the reference standard, the histological assessment. Currently, there's no FDA-approved treatment for NASH. So management strategies include weight loss, exercise, as well as other indications such as type 2 diabetes and obesity indications. These lifestyle changes can be difficult to implement and to sustain. Gastric bypass can reduce the features of NASH and improve insulin sensitivity. However, this is only recommended for those who would qualify for gastric bypass surgery. So, for example, having a BMI of 40 or more. And in a recent study, patients with NASH were identified before surgery and then had a liver biopsy 1 year later. And they found 85% resolution of NASH after 1 year with bypass. However, fibrosis had only improved by 33%. There are some limitations to these management. Vitamin E is also another management strategy. It's an antioxidant that has been shown to have effects on improving NASH. However, there are long-term cardiac safety concerns with vitamin E. So there are a number of drug development targets in development right now, and since NASH is a multifaceted disease, these are divided into the various components such as metabolic syndrome, steatosis, inflammation and apoptosis, as well as fibrosis. And these targets could include anti-obesity and type 2 diabetes medication, addressing the underlying risk factors of the disease. These novel targets of drug development are AMP kinase agonist or FGF21 analogs. For anti-steatosis drugs, the goal is to prevent further fat accumulation, and this includes triglyceride synthesis and de novo lipogenesis inhibitors. Anti-NASH agents are bile acid regulators, chemokine receptor antagonists, or inflammasome inhibitors. And then there are antifibrotic agents. Combination therapy is also being explored to be able to treat the earlier and the later stages of the disease simultaneously. And the NASH indication is a highly active space. There are more than 200 ongoing clinical trials. Many of these have a fast track designation. The most advanced program right now is Intercept's obeticholic acid, followed closely by Genfit's elafibranor. From a regulatory perspective, there are no published guidelines. However, there is a set of recommendations that have been released by the American Association for the Study of Liver Disease in collaboration with the FDA, as well as work done by the Liver Forum, which is a group of industry, academic, regulators, and stakeholders aiming to advance the regulatory science of NASH. And they have proposed the following endpoints for this indication. So for early clinical phase studies, these are typically non-biopsy studies due to the short nature of the phase in the study. So primary endpoints would include safety and tolerability, as well as a reduction in hepatic steatosis. And this is mainly being measured now through magnetic resonance imaging with a proton density fat fraction. There's been much discussion surrounding late phase clinical endpoints, and we now have some consensus that late stage clinical studies will have the endpoints of resolution of NASH with no worsening of fibrosis, at least one stage improvement in liver fibrosis with no worsening of NASH, as well as composite outcomes, such as progression to cirrhosis, liver-related outcomes, and all-cause mortality. NASH is definitely considered the next major health epidemic. There is no FDA-approved treatment, and there are varied success rates with current NASH management strategies, which is compounding this issue. Liver biopsy, which is the reference standard for diagnosing the disease, is not a feasible approach. And to advance this therapeutic indication, there is need for regulatory guidelines, as well as greater general public and primary care awareness. Often patients are being referred to a hepatologist in the very late stages of the disease, and they're missing out on potential lifestyle changes or even opportunities to benefit from clinical trials. And there is need for non-invasive biomarkers for NASH diagnosis, monitoring, and assessing treatment response. And we'll hear a little bit more of this development in this space within the next couple of presentations. So thank you for your attention, and with that, I would like to answer any questions.
For a few questions on the first presentation. I wanted to start one off, Sabina, with just a general question. What do you actually think the first anti-NASH drug label will look like, you know, specifically around what type of patients, and really where do you see that coming from first?
Right, so right now we have the liver biopsy as our main diagnosis method for NASH, and it's definitely going to be prescribed to biopsy-proven NASH patients. Right now, phase 3 clinical studies, inclusion criteria include the NAS score of 4 or more, and they're mainly looking at patients with F2 and F3 fibrosis. So that's more so towards the advanced stages of fibrosis. I believe the labels for these first anti-NASH agents is going to be an F2 and F3 fibrosis rating.
Okay.
I was going to add, once we have more information regarding these non-invasive approaches, perhaps that will change, but for now it's definitely going to be biopsy-based.
That makes sense, and that actually brings me to kind of my next question. We, you know, at High Point, we focus heavily in the proof of concept area, and so does Celerion and other clinical research units as well. And that, you know, we push very heavily to include patients in proof of concept and early clinical phase 1/2A studies. And I mean, the question comes in, does including those patients really contribute more towards just simply the safety and tolerability for the clinical trial protocol, or do you think that there's enough time to actually in the early phase 1-2 studies to actually see any kind of reversion, and not, if not, clearly not resolution, but at least reversion or efficacy effect.
Great question. So this is a long chronic disease, as you mentioned. However, looking at some of these non-invasive measures to track the disease more on the short-term basis, typically what we're seeing from results coming out of the phase 3 studies and phase 2 studies as well is that with about 12 weeks, the time frame to be able to pick up changes in liver fat content by proton density fat fraction.
Okay, so when we're planning out a protocol, that's something we need to be thinking about.
Yes, it's going to be a more long-term type study.
Okay, well, that's actually good information, and we get asked that all the time. So I wanted to see if there were any other questions. I didn't see any come through. So thank you very much, Sabina. We are going to move over to our second speaker, and that's Tim Montgomery. He is the Technology and Market Development Manager at Metabolomics. And Tim heads up the efforts within OWL to bring the understanding of OWL's platform to the clinical development industry. He is highly experienced in launching and promoting novel healthcare solutions, and he'll be sharing how OWL will be able to enrich the selection of the right subjects for the right clinical trials. And Tim, I'd like to turn it over to you now.
Thank you very much, Dr. Vruch. Can you hear me okay and see the screen?
Yep, we can.
Perfect. So thank you for that first excellent overview, Dr. Paglialunga, about the evolution of the NASH disease. One thing I wanted to add to what you showed is very relevant to our audience today, I think many of whom are coming from pharma. Unlike other disease research projects over the years, it's fascinating to see the number of different targets that are being considered with the various pharmaceutical agents under research. I honestly, coming from the pharma industry, I've never seen such a diversity of targets in researching the disease. We do know, however, that reflects the heterogeneity of NASH. There are many factors in disease evolution, and the only common point is the progression of disease and then the ultimate phenotype or the end stage of the disease, which is revealed in the histopathology. And the histopathology, unfortunately, in most of the ongoing studies is somewhat constrained because we're forced to use an invasive liver biopsy. And I think Sabina, in her previous discussion, indicated some of the drawbacks of having to use the liver biopsy as a primary endpoint in the clinical trials. So we all understand quite well the need to find equivalent markers to demonstrate change in disease status. Now there are a number of new technologies available. One older one, of course, is ultrasound. There have been some advancements in that technology, however, that make it more useful. Elastography, MRI, MR elastography, just to name a few. And there's even a genetic marker that's under research that might be useful in patients who have a strong genetic propensity to disease. So a lot of exciting developments. What's important is we need to prove equivalency to the gold standard, which everyone considers to be the invasive liver biopsy. And we've moved along that process, hopefully quite well. And I'm going to show you what our company has been able to bring forward in the last decade or so of research. What Metabolomics, and this is not to be confused with Metabolics. The Metabolomics, of course, is a laboratory technology that is very precise, and we were able to isolate certain circulating analytes that are highly predictive of the presence of fatty liver or NASH, or, as you will see later on, even various stages of disease progression. What you see on your screen on the left are two assays that are commercially available already in Europe. One is for the actual identification of steatosis, fatty liver. Typically, the patient who comes to a liver specialist, the specialist already knows the patient has fatty liver. So that diagnosis is not particularly critical. What's difficult, though, for the specialist is determining whether or not the patient has the presence of NASH, the ballooning, inflammation, stellate cell collection. And this is where the second test, lower left, could be very useful as an adjunct to the other types of technologies used in determining whether or not patient has NASH. I will add at this point that you'll see on the right, we do a lot of work with Pharma already in the NASH space and biotech, and we have close relationships with CROs who are equally involved in NASH research. It's a very exciting area, and I think we're all very fortunate to be part of this amazing area of research. Just a quick overview of how our company got started. A smaller biotech company based in northern Spain. It was a spin-off from academic research about 13 years ago. And the pioneering work was translational using various mouse models. These mice had a propensity of acquiring disease. And what we were able to do was compare the circulating metabolites in these diseased animals with wild type or normal animals, and we were then able to determine, at least in the animal model, which of the analytes change, either rise or fall in value through the course of disease. Now, how can we use these data to begin looking at the thousands of analytes circulating in the human body? So using an animal model, we were able to accelerate the process of looking into human model. And we started out with, in the human, over 540 metabolites, and through versions of the test development, we were able to whittle that down to a reasonable number of metabolites, where you'll see on the far right, the two commercially available tests have approximately 10 to 20 analytes in the algorithm, which is used to determine disease presence or absence. So my point here is it's a very long process. It takes a lot of time. And through this whole process of the human assay development, our guidepost was the invasive liver biopsy. So our studies were done in blind versus invasive liver biopsy. And it's very difficult, as you can imagine, to get a hold of the liver biopsy result contemporaneous with a blood draw. But that's what has to be done to confirm whether or not our test can provide equivalency to an invasive liver biopsy. We also have some really great people who help guide and instruct our research. I think most of these people are known to those on the webcast. At the bottom is the founder, Professor Jose Mato, who incidentally has been involved in liver research for over 30 years. And he's still actively involved in the research areas of our company. Also, we've been very fortunate. I think this year, 2017, we've already brought out a number of important new publications, and this is all the more impressive for a small biotech company. We expect yet a few more in some key journals this year. I won't get into the details of the Metabolomics lab process, but what I would like to point out is it is fairly complicated. It has to be done by people who know what they're doing. And the entire process is very, very precise. This is used to identify unique characteristics of a specific chemical entity circulating in the blood. And you can see on the far right how a certain peak is a marker for or identifies the presence of one specific analyte. I like to show this especially to our very nice pharma clients in research, because we're very frequently asking about the numbers on the left. Okay, you know, how are the triglycerides? What's the LDL doing? And that's all great. But if you look at triglycerides on the left, that value is really a collection of over 110 triglycerides, as you see on the right. So on the left, that's basically the forest, and on the right, those are the individual trees. And when you think about it, if on the right, half of those values were to go up and half of the values were to go down, the overall impact on triglycerides on the left could be 0. But in fact, if you look at it, the individual trees in the forest, there might be significant changes in specific triglycerides in the diseased patient model. So we saw this, I believe, from Dr. Paglialunga. It was a very nice overview of where we are in this whole process of disease diagnosis. Once again, early stage steatosis is typically identified with ultrasound, although in very obese patients, there are issues with the transducer not being able to penetrate abdominal fat, so you get a poor read on the ultrasound. And then above that, the OWLiver is a discriminatory test to determine first if the patient has steatosis, and then whether or not patient is positive for NASH. Very useful clinical test, and I could well imagine when you think about all those other new technologies I mentioned, the elastography, the MRI, those are beautiful technologies, and I believe in the future it will be a combination of those new technologies along with circulating biomarkers. One advantage I would say the circulating biomarkers offer is it can be done anywhere you do a blood draw. You do not need a technologist to run the equipment. You do not need to buy the equipment. You do not need to have a radiologist interpret the result from the equipment. So there are distinct advantages of having a simple early stage, you might even say pre-screening marker test before you move on to the more complicated and more expensive types of technologies. In the most recent and commercially available version of our test, you can see here the ROT curves for both the OWLiver care, which is steatosis diagnosis, and for the NASH test or OWLiver test. The overall ROT curves are between 90 and 95%. And when these data were presented a few years ago, there were many skeptics. They said, this is too good to be true. This can't be real. So we were forced and actually committed to doing validation studies on this information we gathered. And in fact, when we did validation studies, once again, blinded validation studies versus the study cohort on the left, we were able to replicate the results of the test. These are also published data. So we are quite confident that we can offer an alternative in the future to what's considered the gold standard, or invasive liver biopsy. These data are based on a cohort of around 800 patients already. This is all published data. All of these are in verified blinded liver biopsies. And you can see that at the bottom, we are limited though, in that most of our study was limited to the Caucasian population, limited to patients with BMI greater than 25, and only about 20% of those in previous studies were controlled diabetics. There's a lot more work to be done, especially in the area of ethnicity and in patients with various stages of diabetes. We recognize that, and we're working on it and anticipate yet other near-term publications to address these weaknesses. These are some of the studies which are either underway or have been completed, and you can see the splits of patients in terms of ethnicity of the study population and what the percent diabetic in the cohort. So we're moving in the right direction. As everyone knows in research, this does take time. I would point out at the very bottom, always important to recall, all the clinical trials performed are with contemporaneous liver biopsy. What's also exciting is we're underway in looking at the staging of disease, not just diagnosing in a binary fashion, yes, no, patient has disease, but rather, okay, what level of disease, so that ultimately we might be able to determine progression, regression of disease. And this would be extremely important in this patient population because unlike HCV, where you give the patient a drug and essentially they're cured, you do not have long-term monitoring needs. Whereas with all the NASH therapies, these are going to be chronic drugs like hypertension or statins. So they're going to require a minimum of annual follow-ups post-drug launch. And this is the area where an annual liver biopsy is just not going to be practical. And I would venture to say that the patients would reject that concept altogether. Right now in the clinical trials, of course, it's being done because it is an FDA mandate as a primary endpoint in most of the clinical trials, although a few are using MRI and PDFF MRI. So a lot of exciting work yet on the horizon, and we hope that you'll have a chance to follow this closely. I just want to show our friends who are online from the pharma side what we might be able to do and what we actually already are doing in terms of using these tools, these assays. One might be a simple pre-screening to ferret out your patients who are most likely candidates to go on to get a liver biopsy. We know that the NASH trial failure rates are approximately 50% in patient inclusion because they failed a biopsy. And we really believe a simple test like this could help strengthen the overall power of identifying patients who are very likely NASH positive before you send them on for that biopsy and trial inclusion. The other thing is, moving to point 3, the monitoring of drug response to therapy. What's nice about any circulating biomarker is you can do the follow-up testing at will. So if your patient is coming in anyway during the trial for quarterly blood draws, doing an additional test is really no additional burden on the patient or the trial center. So we would urge you to consider that. And then finally, as we move into the area of more specific choice of drugs, right drug for the right patient, personalized medicine, you might consider using these non-invasive tools and technologies to better predict drug response or non-response during the trial phase so that you can present the most robust patient responder group to the regulatory authorities when that time comes. So in closing, some of the partnering strategies that we found very successful is certainly to include these metabolic signatures in your standard clinical trial, because truly the etiology of NASH is a metabolic disorder driven problem. So it makes sense that the metabolic signatures would play a key role in identifying or possibly tracking disease evolution. Also, we believe that you could use these tools in a strategic partnership where you could possibly monitor the patients at more frequent intervals. As it is now in a lot of the trials, patient will start with a baseline biopsy, and then the first real data regarding drug response do not come back until 1 year later when there's a 12-month follow-up invasive liver biopsy. And that's a long time to wait just to get a feel for whether or not the therapy is actually performing as expected. And then finally, I wanted to point out that through our huge database, all of which is biopsy proven, we can actually immediately translate the results of the OWL assays to humans in silico. So we could actually make use of all this data that we've been collecting on patients, both NASH positive and also on patients who are healthy. We've got data on all these different patient groups. So we rely on our database to make predictions about drug reactions. And I also wanted to say, for anybody who is coming to the Washington, D.C. liver meeting in about 2 weeks, we will be present, as we always are at the major liver meetings, and we would welcome to say hello. We do have some presentations. There are other activities. If you're highly interested, please contact me or someone from my company, and we will let you know about some other very specific activities regarding the patient recruitment. So with that, I'll send it to the final slide with my contact information, and I would like to thank you for your patience and your great interest in this fascinating field of medicine. Thank you.
Thanks a lot, Tim. This is Lorraine. We have a few questions for you, so we'll try to be brief before we move on. But we wanted, the first question is a really good kind of preparation question, and it's asking if the metabolomics tests require any type of dietary preparation or restrictions in advance, if beyond fasting.
This is a good question. We have done all-comer studies where patients were on a variety of different drugs at the time of blood draw. The only strict collection requirement is that the patient's blood be drawn in fasting state, which is 8 hours without food or liquid nutrients. So that's the only restriction. What we've also found, interestingly though, is a lot of these specific triglycerides that we have identified as being very relevant in the NASH disease evolution are not especially impacted by diet. So it goes back to that one slide I showed you, the forest and the trees. And in the trees, those 110, many of those that go into our algorithm to make the predictions about disease do not seem to be impacted by diet, as, for instance, the LDL might be impacted if you just had a fat-heavy diet, or as the glucose levels might be impacted if you just had a high-sugar malt shake. So we're kind of protected in that way, but yes, they have to be fasting.
All right, and I have one more question that I wanted to ask you, and that's around developing tests for pediatrics. Are there, as a screening test, do you have any thoughts around doing something like that? And then I have a follow-up question of my own.
Yeah, thank you. Pediatrics is a huge, important area, and we've had a number of discussions with high-level pediatric hepatology centers. One of our close informants, I would say, is Miriam Voss from Emory University in Atlanta. She's very well known in the circles, and she understands this technology and the value it might present to the pediatric population. One challenge is getting the biopsy matched samples. It's incredibly difficult to find a contemporaneous biopsy and blood sample that have been preserved So we're working on that. We realize how important it is, but at this point, it's second stage or second fiddle to the adult model. And I would add that the current trials underway, to the best of my knowledge, they're all adult trials. So we're kind of teaming up with what Pharma is looking at right now, and that is adult NASH.
All right, well that actually, I'll just make the comment and we can talk about it at the end. And maybe there's something already out there, but it does make me wonder if there isn't room for the industry to work together to use some of the biopsy material that they're collecting along the way to really create sort of a more of an open forum database through a technology like this, and maybe we'll talk about that later. And so we're going to go ahead.
That's an excellent point you raised, and we on one of the slides I mentioned the scientific collaborations. You know, we're a small company, but we are always looking for opportunities to collaborate, and this would be an excellent way of collaborating with groups. We are having a meeting with some people who have a focus on pediatrics at the upcoming liver meeting. But any other pharma companies that might have an interest in this, please contact us, because we might be able to come up with some study design that benefits not only the pharma company and us, but most importantly, benefits the children.
Agreed. All right, well, thank you very much, Tim. That was really interesting. And we'll hold any of the other questions for the end of the panel. It actually flows really well because speaker number 3 is our own Dr. Margarita Nunez. She's the High Point Clinical Trial Center medical director, and she's actually working on a specialized project, and we're actually looking at doing some additional enriching and pre-screening of our database just along the lines of Tim's technology, and really considering it in addition to the FibroScan technology that we've activated and begun using to really screen our own volunteers and patients. So I'm really excited that Dr. Nunez is able to join us. She's, as I said, our medical director and PI. She's got over 20 years of clinical research experience and completed more than 450 trials. She's practiced internal medicine, and as our medical director at High Point, she really does head up the special patient population studies, and one of her current remits is becoming a NASH expert in early clinical trial development. And today she will review the recruitment, diagnosis, and medical management of these patients in the early clinical development studies. So I'm going to hand it over to Dr. Nunez.
Thank you, Dr. Rush, for that introduction, and thank you for the 2 previous speakers. Very informative slides, and I sure hope that we can develop some of these partnerships and work with some of these non-invasive biomarkers, because as I'm going to describe over the next few minutes, it's just the challenges that come with recruiting an illness such as NASH. So here's my life. I'm the medical director, as Dr. Rush said, and I will be discussing effective patient outreach and some of the recruitment strategies that we've implemented here at High Point. So I want to highlight some of the challenges with diagnosis that have already been alluded to previously, but how they impact identifying and recruiting the patients, discuss some of the specific site perspective and demographics here in our area, and discuss some of the more innovative outreach strategies that we're developing here at High Point. So what are the challenges with diagnosis? I know that Dr. Paglialunga already alluded to some of them, but when you think about effective patient outreach, you have to first start with how to identify the disease that, as we discussed, although it is one of the most common liver disorders in the Western industrialized countries, and it seems like throughout the world, there's very little awareness of it in the general population. Most patients with NASH and NAFLD are asymptomatic. Actually, mostly NAFLD are asymptomatic, and even those with NASH can have very vague symptoms, unless they obviously present with stigmata of hepatic cirrhosis. Fatty liver is often an incidental finding on abdominal imaging that is often performed while treating unrelated conditions. The subjects, as mentioned previously, may have normal or fluctuating transaminases, and there are no well-established non-invasive methods for diagnosis, and the gold standard is still liver biopsy, which is not a common practice due to the cost, the low cost-benefit ratio, and in reality, not having an FDA-approved treatment just diminishes the risk-benefit for that procedure. So how do we overcome then these challenges? We know what the major risk factors for NAFLD are. We know that it's central obesity, type 2 diabetes, and even insulin resistance, dyslipidemia, metabolic syndrome. There are also associated illnesses like systemic hypertension, and specifically with metabolic syndrome, there is an association with an increased risk of more severe fibrosis. It is also very important that other disorders be ruled out, and during in a clinical research setting, although we screen for these other illnesses like hepatitis, this particular hepatitis C, since the demographics of the people who have NASH seem to be kind of similar age group of the prevalence of hepatitis C in our community right now. And of course, there's being able to rule out and getting a close medical history to rule out alcohol abuse. So that's why understanding the fibrosis, fluctuation in liver enzymes, as well as the connections to diabetes and obesity are in reality the keys that were prompting our methods that we're coming up with identifying and recruiting these patients. So the answer is simple. We have to go to the risk factors because that is the simplest way to do it on a large-scale basis. We basically try to go where the patients are. We need to raise awareness and educate patients and the community as a whole. We know NAFLD and NASH can be life-threatening, and information can guide patient care and motivate them to enact lifestyle modifications. Patients who are found to have cirrhosis will require screening for esophageal varices, whereas patients with early fibrosis can then be motivated to lose weight in order to prevent cirrhosis. So what are some specific site perspectives and demographics? It is important to recruit effectively and eliminate as many logistical issues as possible. As I mentioned before, we try to go where the patients are. One of the first programs that we've established here is partnering with a local community clinic, volunteering to provide care benefits, obviously the patients. It really helps everyone. It provides opportunities for education, informing the patients about ongoing clinical research, and bringing to them the opportunity to participate in a study where they may benefit. It increases the visibility of the clinic, and it helps us earn the trust of the community and developing relationships from the beginning with potential subjects. So the relationship between the investigator and the subjects in a condition like this is very important in all the metabolic disorders in reality because lifestyle changes are so key to the management. It is very important to know your community. We know that the prevalence of obesity is very high in North Carolina, and lifestyle modifications that we often recommend are in conflict with cultural norms. However, positive recruitment outcomes are closely related to the level of trust and strength of relationships between the clinical site and the community. So what is our rationale and the strategy that we're using? Again, we know the progression of diabetes and obesity leads to a creation of a subset of patients with pre-NASH and NASH. We have over 15,000 patients in our database with diabetes and obesity. We have a data-driven outreach based on the inclusion-exclusion criteria for several studies that we can easily match. But proactive screening for risk factors of NAFLD and NASH can be done very simply. I have here BMI, diabetes, and even just something as simple as waist circumference. And you add all of that to your database, and then you know how to reach these subjects. We have formed strategic alliances with local gastroenterologists and hepatology practices who are interested in serving as sub-investigators on our trial, and some of them just interested in referring their subjects to us because this is a condition where there are very few treatment options. So offering the opportunity to participate in some of these trials is really understated. We have recently invested in our own in-house fibroscan. As it was mentioned previously, we need more non-invasive ways of identifying the disease, and this is the transient elastography is often used now in some clinical trials. So by having our own staff trained in how to conduct these and offering this as a pre-screen tool for some of our diabetic subjects has been very helpful in identifying patients with NASH. By measuring the lack of elasticity in the liver in a non-invasive way, it can be used then to estimate the severity of liver fibrosis in NASH. So what is another specific HPCTC approach? One of the keys to successful recruitment has been to start with a recruitment team that can have very positive interactions with subjects from first contact. Our specific recruitment team is in-house. They're employees, GCP trained, comfortable with patient-centric care, and they understand the patient profiles and conditions. They participate in site initiation visits and internal meetings. They maintain daily discussions with investigators and coordinators, and they can highlight the importance of compliance and adherence to protocols before the subject walks in the door. They really give them a little bit of our culture while they're on the phone with the potential subjects. They're also able to address questions and alleviate social cultural fears. We heard earlier that NASH is prevalent in the Hispanic population. The numbers that I had seen are in the patients with NASH, about 45% Hispanic compared to 33% whites and 24% blacks. So we have a bilingual team, and we're translated into Spanish so we can attend some of the community events where those subjects will be. So what are some of the community initiatives? By setting forth some of these initiatives, it helps us identify the patients. As I mentioned earlier, the medical volunteering at a community clinic. We offer to do free screenings even at those clinics where the patients, they usually lack a lot of resources and the patients are not usually aware of the opportunities for research. We've made a significant investment in the FibroScan that has become very, very helpful. And the participation in health fairs, this is one of the things that, it's not just health fairs, but in reality, festivals and community events in general. We recently had a 4th of July, just general citywide festival where we were very present, and we have a mobile recruitment unit that we can take around to different rural communities, and we use it to perform some of these simple things like calculating BMI, doing blood pressures, and even point of care things like hemoglobin A1Cs and glucose at these events. And then we hand out information about the potential diseases and some of the IRB approved things that we have about the particular studies. So what are some of the positive outcomes of this? What are some of the results and the benefits of all these efforts? With High Point Clinical Trials' continuous commitment towards the community, it really opens the door for increased interactions with patients. We have identified through some of these outreach, since we started, about 500 plus diabetic subjects and hypertensive in just the last few months. And then, of course, pre-screening can lead to identifying several subpopulations like NASH, but also some associated conditions like chronic kidney disease, hepatic impairment, among others, which will allow other drugs to be tested in those different populations as well. Access to research offers options to patients and a better understanding of their illness. Early diagnosis and recognition of illness leads to early intervention and better outcomes. So, by engaging the community and establishing a relationship of trust with patients as research partners, we can lead them to better outcomes for the subjects, as well as efficient enrollment in clinical trials, obviously leading to better data, and ultimately leading to treatment options that are very much lacking for this condition. So in conclusion, we are partners representing the standard of care while contributing to strategic planning and execution of clinical studies. But in reality, it's the partnerships, not only with the community, with the patients, and like with speakers who are also on this panel, that will ultimately lead to the best treatment possible for this very prevalent condition. So it should be a win-win for all. And that's all I have. Thank you so much for your attention. And if you have any questions, please do not hesitate to ask.
Hi, Dr. Nunez. That was really great. Thank you. I think it helps us to just remember that it's one thing to design a protocol, it's another thing to get the patients and then medically manage them. So your presentation, I think, started to help people think about really what type of protocol and how do we get the people that are going to be best fits for these studies. We had one question out there that was around the around subjects that have NASH, and what percentage of those subjects actually may have hyperinsulinemia.
So I don't know if Sabina has a number specifically for that, but I don't have one right now, but I can tell you that in everything that I have seen, it's not just diabetes. I know that we're identifying diabetes as a precursor for NASH, and most of the protocols that I have seen require the subjects to be diabetic as a risk factor unless they have a biopsy-proven NASH. But it's also very present in just insulin resistance. So very early on, we're kind of missing some of these with these patients by not screening them a lot earlier.
And so, I mean, I think that actually goes to our pre-screening type of activities. One of the questions I had, and it's something that, we deal with as we get the protocols, and it's really kind of late, late in the process, so I wanted to bring it up here, is, we deal with inclusion-exclusion criteria as we're looking at feasibility assessments on protocols. And in your opinion, we're starting to see more and more protocols in early clinical development, but from what you've been seeing in the limited number of protocols that have come in already, what do you find is the most challenging inclusion-exclusion criteria for some of the early NASH studies, in your opinion?
Well, I have two. One of them is obviously the requirement for a biopsy, because not everybody comes in with a biopsy, and in reality, it's a more difficult thing to convince somebody to do. And like my colleagues were saying earlier, even trying to follow those throughout the trials, it's harder to convince somebody to go through this more risky, invasive procedure. So that is one of them. And the other one is, as I said before, and as the person who asked the question probably alluded to, we're probably missing a lot of subjects by focusing mostly on diabetics. I think that it comes much earlier than that. As prevalent as type 2 diabetes is, it's still underdiagnosed. Just last week, I spoke with a subject who came in for a healthy study. She said she had gestational diabetes years before. Her last A1C was 5, and when I went to look at her labs, her A1C was 12. And so even though diabetes is very prevalent, we have lots of medications out there, and there's a lot of research still going on for diabetes, we're still kind of missing those very early people who, for one reason or another, whether it's for lack of education, understanding, the physician not having the time to explain the significance of it. I think by requiring patients to be diabetic, they're going to miss a lot of subjects who may just be obese, who may just have insulin resistance and already developing fat infiltration of their liver.
So do you think that basically HbA1c and maybe a FibroScan or some type of other confirmatory test might be something to consider as a separate, a substitute inclusion for just diabetes?
Definitely the A1C and the FibroScan, but what I'm saying is that we may be missing the ones even with the lower A1Cs who still have fluctuating insulin resistance, but maybe already be developing. Like, I don't know exactly at what level with the subjects start developing the fat infiltration, but I think with earlier, looking at earlier evolution of the diabetic spectrum is going to be necessary.
Okay, well, thank you very much, Dr. Nunez. And I would just add to that, Dr. Nunez has been working with our local hepatologist and gastroenterologist, and even with the largest database in North Carolina of patients, of NASH patients, the first comment and the shocking reality of it is that only 1% of that type of database is actually biopsy confirmed. And so even the specialists really don't have those subjects in their database. And when we discussed it, the feedback was, well, why am I going to, put a subject through that for a diagnosis where there's no therapy? So really, biopsy is just not a realistic way to support the industry's need to do clinical trial research, at least in the early stage. So I think that goes back to alternate mechanisms and means. And so with that moves us over into speaker number 4, and that is actually Dr. Michael Murphy. He is the Pacific Biomarkers, he's from Pacific Biomarkers in Seattle, Washington. Michael is the lab director and strategic relationships manager at PacBio. PacBio is a leading developer of biomarkers utilized in the clinical development environment. He's going to share their thought process around the future of effective biomarker tools, how to develop them in a systemic environment, and what the latest biomarkers under evaluation in the clinical setting is. So with that, I want to hand it over to Michael.
Thanks, Lorraine. I appreciate the intro. And I also have to say that I really appreciate the content and presentations of the other presenters. I think this has been a really nicely balanced webinar so far. And for introduction purposes, maybe a number of people may not be familiar with Pacific Biomarkers. We're actually a 28-year-old expert, specialty biomarker provider for servicing drug development. And our primary focus and expertise is in and around the ligand binding assay performance. And so things such as standardization, validation, optimizing assays, and critical reagent method maintenance and oversight in QC have been really key things for us in the long run. Also, historically, we sprang out of the University of Washington Medical School back at the beginning. And the focus, and I think it's particularly germane to this topic, the focus at that point was heavily on cardiovascular disease. We were an original, one of the charter members of the CTC group that was charged with standardizing lipid testing, actually on a global basis. And from that emphasis in cardiovascular disease, our interests have continued to spread in the areas of metabolic diseases such as diabetes, metabolic syndrome, and obesity, as well as logically as that goes into liver disease and NASH. And likewise, inflammation is also a key element here, and also organ injury. We've had a lot of emphasis in recent time in acute kidney injury. And basically, to kind of sum us up in a snapshot, we're a CAP-CLIA accredited lab with considerable bioanalytical expertise. And what I tell sponsors and so forth, that we cover the biomarker continuum in a unique way. And our clinical experience and bioanalytical experience really come into play when you look at diseases such as NASH and liver disease, and other metabolic diseases. We see that plays very squarely to our strength. My disclosure. What I'm going to cover, I'm trying to minimize the amount of redundancy that from the previous presentations, and kind of focus on those elements in and around soluble biomarker relevance and standardization, and maybe a few different perspectives as they relate to liver biopsy, and then finish up with some examples of biomarker data in published literature, and then some current biomarkers in use from aggregate data that I've collected from our laboratory in presumably NASH subjects as part of studies that we've been involved with. Now, as previously mentioned, NAFLD is a most common chronic liver disease with a prevalence roughly of 10 to 40% in Western countries. And the other thing that I would add here that I don't think was mentioned before, but the projected number of individuals with NAFLD in the U.S. is approximately 65 million people at a direct medical cost of $103 billion per year and growing. And as was mentioned before, because of the success in dealing with hepatitis C, we do see and expect that NASH will take over the leading reason for liver transplantation in and around 2020. NAFLD remains amongst the most common liver diseases worldwide, with prevalence in concert with increasing prevalence in concert with obesity, diabetes, and metabolic syndrome. Our understanding of the natural history of NAFLD is constantly evolving, with nascent data challenging current dogma. There remains some ambiguity regarding long-term outcomes in NAFLD, with some conflicting data observed amongst various studies of the natural history. Certain subjects in NAFLD may be progressive, while fibrosis remains among the most important predictors of hard long-term endpoints, such as mortality and liver complications. Identifying the predictors of long-term outcomes should be used to direct development of clinical trial endpoints in NAFLD. Now, I want to make a few comments in addition to what's been mentioned about biopsy. People do question the term gold standard versus reference method, and with some justification. But the fact of the matter is NASH is defined by histology. It is a surrogate primary endpoint in clinical trials, and it remains central in the field and will for quite a long time. Variability, morbidity, and mortality risks, patient compliance, and limited workforce capacity all bring into question the large-scale application of liver biopsy in clinical trial. Efforts are being made to improve the comparison between biopsy and new and existing soluble biomarkers. There's also efforts in the field to help deal with some of the alienarity involved in liver biopsy histopathology. And those efforts, I think, will make an impact and will help further the development and characterization of non-soluble or soluble biomarkers. But, as we look at the impact of biopsy-based approach on drug development, it really breaks down into how it impacts screening. And when you look at the large number of biopsies that end up being, that are not collected either properly or have questionable collection, it's quite large. The impact on recruitment obviously is telling, because either people may not want to enter into a clinical trial because of the discomfort and issues around biopsy, but certainly recollection is not something to be looked at favorably. Also, with some of the issues and challenges, it brings into question some of the decision-making and timeliness for clinical trials if you're constantly dealing with those kind of issues. And this could result in potentially very good drugs being developed and being and brought to market, but the decision-making process relative to molecule or dose selection may have been impacted by the results from biopsy observations. And then as far as the clinical translation, you could have a perfectly well-developed drug, and if correct, at-risk patients are not identified in clinical practice in a timely manner, the drug's impact in terms of release burden could be. Now, I want to make a few additional comments about the pathophysiology shown in this slide. As was mentioned before, excess dietary saturated fat intake, as well as increased adipose tissue lipolysis, contributes to free fatty acids in circulation, with increased fat accumulation in the liver. Overconsumption of fructose and dietary saturated fat, leading to increased de novo lipogenesis, also adds to hepatic steatosis. Insulin resistance and oxidative stress fuel a lipoxic environment within the liver, which can result in hepatic inflammation and injury, leading to fibrosis. In light of the other diseases that seem to be increasing in concert, combined therapies addressing other conditions such as diabetes, metabolic syndrome, obesity, and cardiovascular disease may evolve as a key therapy approach given the diversity of NAFLD-NASH. Now on this slide, I'm showing a kind of a high-level progression of the natural history of NAFLD-NASH and onto hepatocellular carcinoma. But in looking at these various opportunities for progression or risk assessment or whatever, biomarkers that diagnose relevant information about progression, risk, staging, or stratification and response are going to be, continue to be explored and mentioned, as mentioned before in other presentations. Those procedures, depending on which part of the spectrum you're focusing on, we can see that early on there are methodologies that will contribute to maybe identifying people at risk for progressing onto more advanced stages, as well as characterizing folks that may have other predisposing situations that they should be watched over time. But as progression continues, other markers are needed and need to be characterized. And again, well-designed studies, well-defined cohorts also need to be identified in order to successfully further characterize the impact that these methods can have. And then in later stages, again, having a good idea of any possible improvements, whether to inflammation or fibrosis, would be very helpful. And then also just examining and being aware from a clinical standpoint whether or not the cirrhosis and or eventual hepatocellular carcinoma in those cases, what options might be seen given with future therapies that arise. So why do we need biomarkers? Well, kind of listed some key reasons why the biomarkers are needed and broken them down to identification, response to therapy, and drug development. In the case of identification, certainly identifying individuals at risk for NASH, individuals who may have simple steatosis, and those that may not, but being able to better characterize and identify would be very helpful, both clinically and from a clinical trial standpoint. Individuals at risk for liver fibrosis due to NASH, identifying them, as well as individuals with fibrosis. Earlier stages, the information is much harder to get at, but hopefully we'll be able to better characterize throughout the entire spectrum. And again, that goes back to devising ways to help histopathology become maybe a more linear measure of tissue damage. I'd also mention that being able to diagnose these individuals as early as possible would make for a very positive impact. Response to therapy, resolution of NASH and fibrosis, and or improvement or resolution of either or. This will be very important because there may be therapies out there that, again, with the combination approach, it'll be important to identify maybe improvement in inflammation versus fibrosis and vice versa. And then drug development, screening techniques for NASH and fibrosis, as well as detecting subtle or significant changes in NASH and fibrosis would be very important. The typical areas of interest and biomarker usage are shown in this slide as it relates to NASH. I've listed here, this is certainly not a comprehensive listing, but I've listed a number of various soluble biomarker assays that are being used to evaluate and to try to evaluate the response to methods or to therapies that are in clinical trial. and they are continuing to do more and more work to increase the knowledge base and impact of these biomarkers. What I'm showing you here is a table of some key liver injury markers. There's a wide range of biomarkers and panels that have been tested. In general, studies are small and need further validation. The sensitivity, specificity, and the area under the receiver operating characteristic curves may vary depending on the defining of the cutoff point. So in this list here, I highlighted a couple of biomarkers that show good promise with good ROC information at various cutoffs. And again, those are somewhat study specific and cohort specific. And then one example for a key liver panel that I wanted to show here is with the ELF. And at the given cutoff for the ELF, in this case, the ROC looked very good. What really sensitivity and specificity are indeed important, but knowing what the characterization of your cohort population and the incidence of diseased individuals can play a big role in the way that your biomarker or liver panel can perform. And so I think this information in terms of positive predictive and negative predictive value has a very important role in interpreting information. Now there's also, I wanted to mention something about the standardization efforts. FNIH has a program currently underway that is called NIMBLE, standing for Non-Invasive Biomarkers of Metabolic Diseases. This is an effort to identify, standardize, and advance qualification of set biomarkers fit for purpose, non-invasive biomarkers such as circulating, functional, and imaging for diagnosing and staging of NASH. And this is in individuals that are at risk of progression to adverse outcomes and in need of pharmacological intervention or non-pharmacological intervention, or assess risk for progression and response to intervention in subjects with various stages of NASH at baseline. There's currently 2 stages here, the first stage being a large retrospective analysis of current non-invasive NASH biomarkers, followed by a second large prospective study with the circulating functional and imaging biomarkers in an effort to qualify the biomarkers for use in clinical trial. The second half of that stage 2 will presumably be used in conjunction with the therapeutic agent. And there are other registry types approaches for gathering more information over long longitudinal periods of time, such as the litmus liver investigation, as well as target NASH. Again, the idea is to identify over time clinically meaningful endpoints. Now, some published information I want to show, just to give some examples of the kinds of biomarker identification and characterization that's underway. Is these are 2 examples with GLDH and MDH correlated against alanine, the alanine transferase and ALT. And both of these show good correlation with increasing levels of ALT, and so they have some continued possible uses in certain cases of clinical trial and diagnostic use. And then below is the ROC curve showing that those 2 same biomarkers show good ROC performance of the assay in comparison to the other 2. This is a little different perspective where alpha GST, again, identifying the purpose and the use of the biomarker relative to the particular study. In this case, looking at acetaminophen poisoning, the example here is that there was a need for a more immediate indication of a more acute indication of improvement or further damage relative to traditional transaminase markers here. And then lastly, I wanted to show an example here of triacylglycerol. And in this case, TAG1 was significantly increased in NASH patients compared with that in non-NASH subjects in both the NAFLD and the chronic hepatitis B group. And you can see that over here. The ROC analysis for detection of NASH and NAFLD group showed that TAG1 presented good diagnostic value similar to that of CK fragment 18 M30, and that was better than both the M65 and ALT. And so again, depending on the cutoff used, you can see differences in how the performance of the biomarker may be given the specific situation. Now, just to go through a few examples of data, aggregate data that I've collected from a number of cases in our laboratory. And basically, in this case, P3NP, which is a fibrosis marker. And if you set at a literature cutoff point here, you can see that presumably these are individuals from a host of trials that are showing elevations in the P3NP, and I would assume that a large majority of these folks are NASH patients. Total adiponectin, again, the cutoff here is set by one of the literature references. And again, you see a high concentration of lower adiponectin, which is the result of what you see typically in insulin resistance and possible diabetes. And so again, these are a lot of these are NASH patients at various stages. Hyaluronic acid is another fibrosis indicator, and again, set with a cutoff set at this level, we see a good number of the NASH subjects being presented with higher hyaluronic acid values. And then lastly, the CK18 M30, again with a cutoff set here, we see a good number of subjects in NASH studies that are presenting with fairly high values. So in summary, NAFLD NASH is a complex disease entity with considerable unmet need. There is a need for effective biomarker tools to support professionals involved with the delivery of care and development of effective therapies for NAFLD NASH. New biomarker tools need to be developed in a systematic and standardized manner, such as those, the comments I made about the NIMBLE group. And then new soluble biomarkers continue to be identified and explored in an aggressive manner, and more with both the involvement of academia and industry in a collaborative manner. And with that, I thank you for your attention.
Great, thank you very much, Michael. That was really interesting. I have a question, but I wanted to address one of the audience questions. And okay. That question is, do you work with genetic markers or SNPs or methylation patterns for diagnosis or prognosis among metabolomics?
We do not. We are not currently involved with any genetic markers or techniques or platforms at this time. It is something we've talked about branching into, but have not done it to date.
Okay, and you know, and maybe I don't know if Tim can pop in when we're done here, but I had a more of a discussion / question around the biomarkers in general. I mean, a lot of these are used in the information gathering stage early in clinical development, and then they, you know, tend to be better data or more interesting data and become endpoints. But the question is, and you know, the FDA has been, I think, clear but somewhat ignored in its 2013 guidance, saying that basically if you use a biomarker, it should be validated where it's supporting a claim or it's an endpoint or it's, you know, in the realm of on the decision spectrum. And so most of these biomarkers right now are still in that CLIA stage. And how do you work with clients on the spectrum between a CLIA assay through a qualified assay into a fully validated GLP assay, and what are your thoughts around those different tiers of validation and compliance?
So Lorraine, this is Tim. I was listening in, and I didn't address this specifically in the previous presentation, but indeed there is an interest, and we are working with some pharma clients on developing a companion diagnostic, a CDX, which has to go through a different approval process. It's a non-CLIA. It's not a LDT, laboratory developed test, but rather it's a in vitro diagnostic. Goes through a 510K or a PMA process with the FDA. So on those specific situations where the company would want to pursue a specific companion for their drug, which would quite accurately predict response, non-response, that has to go through the FDA process, and we partner with people on that level as well. It's more complicated, but in the long run, it goes back to that more personalized approach to therapeutics, where you have the right therapeutic for the right patient.
Okay, I think that answers the earlier question, correct?
Right. I'd like to comment on the way we have seen the validation process has been, the work that's being done, for instance, with the Nimble group takes into account the need to identify retrospectively a host of biomarkers that are available, and to pick in a very precise a manner as you can, which ones will you move forward to obtain context of use and further qualification status via the FDA. And that's a fairly lengthy process involving both the well characterization and standardization of assays, and then relating them back to reference methods or gold standards. But for a lot of the work that we see, the CAP-CLIA versus the bioanalytical approaches, we see a mixture where very often in our case we will at least approach validation work from a CAP-CLIA standpoint, and then depending on the what is the fit for purpose of the particular biomarker in a given circumstances, we will apply the appropriate regulatory compliances as appropriate. So many times when we're engaged in and asked by a sponsor to assist at the exploratory level, we will gauge that process, and I think transparency and communication, especially as you're moving through this entire continuum, is extremely important. So it is important to get it right, to establish a good stake in the ground in order to reference back as you move through this process, because there's always that concern about you don't want to have a biomarker failure due to something that was neglected early on. So again, and you're dealing with a very complex disease entity, and that's we've seen things happen in other instances that are related to what you see in NASH and liver disease.
So Lorraine and Dr. Murphy, I would like to compliment Dr. Murphy on his great overview, because he hit the nail on the head in terms of standardization requirements moving forward for some of these new biomarkers. I did not mention the fact that the OWL Metabolomics assays are part of the Nimble, and they are part of the Litmus trial in Europe. So we were very thankful to be included in that process. As we understand it, this whole process of evaluating and standardizing the various markers could take some time. So we're patient with that, but we also realize that the individual companies in their clinical research requirements need something nearer term. So it would probably be wise to consider the long-term benefits, but also think about what can we do to increase the likelihood of prediction by using new or alternative tools beside the simple liver biopsy.
Agreed.
If I may, one last thing I would like to mention is there was a question that was asked of Tim after his presentation about diet influencing the kind of testing that his company is doing. And I think in a broader point is we can't underestimate the standardization goes from pre-analytical to post-analytical considerations. So those pre-analytical considerations, whether it's stability of the sample, additives, timing, diet, and so forth, very important as well.
Yeah, and they just continue to add up the variables and contribute to the noise, right? So I think that a lot of times, even when we're looking at the data, the biomarker data, we're not necessarily thinking about that, and we're looking for these very minute changes and trying to impart meaning when we forget all of the variables that have added up to make that almost impossible. So I wanted to mention that we have opened up the raise hands function, that if anybody wanted to go ahead and raise their hands, we'll be able to actually take those questions as we're seeing it. And while people are thinking about that, I wanted to actually open up the panel and ask the panelists. Tim and Michael have been doing that so far, but if Margarita and Sabina had any additional questions that they wanted to ask the panel or come back and share their thoughts, this would be a great time to do that as well. And I believe the mics are open.
This is Dr. Nunez. I really just want to say that it was so informative to have them here, and I'm really looking forward to seeing some of the upcoming biomarkers so that we can screen subjects in a different way. But this highlights the importance of this type of webinar and education outreach to our whole community. I mean, and this is I'm assuming the audience is kind of part of the choir as well. But we need to get this message out to the medical community in general so that we can come up with more effective treatments and screening mechanisms.
Yeah, I actually like the idea that we're able to bring people from all different avenues of clinical trial research and therapeutics together in one setting, and we'll probably be looking at ways to expand that in the future, but yet still in a digital electronic way so that people all over the globe can actually attend. But it's really important to not develop these therapeutics in a silo, meaning the bioanalytical biomarker people can't be off in their own silo, the physicians in their own, and the drug development scientists in their own. So doing it early now and developing it more as a community, I think is only going to be beneficial. And I really do hope that maybe in one of the liver-centric focus organizations, we can talk about what a consortium for using data from biopsies actually looks like, because again, there's only so much of that to go around, and it really freeing up that information for everybody to use really only helps everyone's individual drug development program along the way. And I think we have to work towards a more collaborative type of environment as opposed to sort of the very intense competitive environment that we've all had. And especially this is a great opportunity because NASH has so many different targets and so many ways of interacting that I think it's really a truly beneficial way to look at things.
Lorraine, this is Tim again. There is a group that is academically driven, if anyone is interested. It's called the Liver Forum, and they're out of Berkeley. They have been traditionally very involved in liver-related diseases, beginning with HIV and HCV. Now a lot of their focus is NASH. So I would urge anyone who's interested in exactly what you mentioned about this intradisciplinary collaboration to look up Google the Liver Forum out of Berkeley. They're just a great group of people, and they have a lot of follow-on from the pharma industry.
Okay, that's a great suggestion. We have a question that came in that's marked for Dr. Nunez, but it may actually also apply to Sabina. The question, if I read it correctly, is biologically, NASH is an anabolic disease with meaning too many nutrients that the liver cannot handle. Were there any comments on that? It's a little, does anybody have any comments on that?
This is Dr. Nunez. I don't have a specific comment other than, I guess we just go by what we know histologically, and we know kind of historically about this disease. We know that, the effect of fructose. We know, I don't know about nutrients in general. I have not seen that specific, a study specific to that. But we know that diet and exercise is the only effective treatment. And what that means is they still, things like Mediterranean diet are still recommended even for something like NASH. So I don't know if Sabina has any comments specifically that she's come up with in her research.
And just to be clear, it says nutritions, but I'm assuming they mean nutrients. So I could be wrong, but I think it means nutrients.
In general, it kind of leads me to think it's excess calories, which we all know, even from, lab rats, that, an excess calories is really kind of what affects a lot of illnesses. And NASH is just kind of the epitome of that.
Yeah, this is Sabina. I'd like to echo that, and how definitely diet, exercise can go a long way with reducing both the elements of NASH itself as well as fibrosis. And even current management strategies suggest that caloric restriction could also help with lifestyle changes for managing NASH.
Okay, and here's another question. It's a biomarker question, but it's also a little general. It says, have you seen glucagon elevation in the biomarker studies? Go ahead, Michael.
Okay. It seems to me I can't say one way or the other, to be honest, but we have seen studies that we have been involved with where glucagon has been included. My inclination would be that we probably are seeing some elevation in glucagon, but I haven't specifically looked at any of our data where we have generated glucagon, but I certainly, if that question has come in, I can certainly research that and get back to that person.
Okay, and Sabina, didn't you look at some of this in some of your work, I thought?
Regarding the effect of glucagon, I would need to go back and look at that, but there definitely is effects once you're changing insulin sensitivity. We have effects on insulin as well as glucagon, so there is potential that there are changes there.
Okay, well, I think right now that hits all of the questions. Hold on. I'm just double checking to see if we have any more questions to go through, but I think we've gone through them. And I wanted to take the opportunity to thank the panel. I think it was really nicely done and everyone really kept to their time. We will, of course, be following up with a formal document that just, whoa, a giant question just came in. And since we're all still on the line, let's go ahead and do it. The question is, NASH biology is still being explored and will lead to different data to collect and endpoints in the years to come. There is a risk to sponsors. For sponsors is that their ongoing clinical study, which can take years to complete given limited enrollment rate, including competition for patients, i.e., their data might become obsolete. How are the regulatory authorities going to face this issue? Sabina, I think you were doing a lot of the early kind of regulatory looking to see, and I have my opinion, but I'd like to hear if anybody wanted to step up and take a swing at this question.
Yes, Sabina, I can definitely start. So a lot of these agents right now have a fast track designation. And they tend to have a 72-week interim analysis, in which this mean fast track designation will be decided upon. So although the biomarker space is advancing and advancing rapidly, I don't foresee that there is a risk at the moment for this to be a significant issue in drug development.
Okay, and I mean, I think I agree with you. I think that, it's as long as any other drug development program is to enroll, and we're always at the risk of the regulators' change of thought. But if your clients are having regular contact, regular documented contact with the FDA, and you include an interim analysis and the plans are agreed upon ahead of time, that's about as much as you can do for this or any other drug development program. So with that, we've actually gone up to about 2 hours. And so I wanted to say thank you for everybody for joining. Thank you for the presenters. You did an excellent job. We will be pulling together the recording of the webinar and making that available. And we will be following up with the fact sheet and everyone's contact information so that everyone on the webinar listening can reach out directly for additional, more in-depth discussions. We don't want to get in the way of that. We want to give you free access to the experts. That's what we're doing this for. And I wanted to thank the audience for hanging in with us. There's still about 64 people on the line, which is not a bad showing. So again, everyone, thank you and good night.